1. Which of the following components is unique to the cell wall of fungi, distinguishing it from plants and bacteria? A. Cellulose. B. Chitin. Correct. C. Peptidoglycan. D. Lignin. Answer key, B. Explanation. Fungal cell walls are uniquely composed of chitin, a polymer of N-acetylglucosamine. Cellulose is found in plants, peptidoglycan is found in bacteria, and lignin is found in the secondary cell walls of woody plants. 2. According to the fluid mosaic model, what is the primary role of cholesterol in animal cell membranes? A. To act as a receptor for water-soluble hormones. B. To regulate membrane fluidity at varying temperatures. Correct. C. To facilitate active transport of ions. D. To form the basic bilayer barrier against water. Answer key, B. Explanation. Cholesterol acts as a fluidity buffer. It stabilizes the membrane, preventing it from becoming too fluid at high temperatures, and preventing it from freezing, or becoming too rigid at lower temperatures. 3. During cell fractionation, which organelle sediments first at the lowest centrifugal speed due to its large size and high density? A. Mitochondria. B. Ribosomes. C. Nucleus. Correct. D. Chloroplasts. Answer key, C. Explanation. The nucleus is the largest and heaviest organelle in the eukaryotic cell, so it pellets out first at low speeds, typically around 601,000 times g. 4. Which eukaryotic ribosomal subunits combine together to form a functional ATS ribosome? A. 50S and 30S. B. 60S and 40S. Correct. C. 60S and 30S. D. 50S and 40S. Answer key, B. Explanation. Eukaryotic cytoplasmic ribosomes are ATS, consisting of a large 60S subunit and a small 40S subunit. The 50S and 30S subunits combine to form prokaryotic 70S ribosomes. 5. A congenital defect or absence of specific lysosomal enzymes results in the accumulation of macromolecular substrates inside the cell. These conditions are collectively known as A. Metabolic oxidation disorders. B. Lysosomal storage diseases. Correct. C. Secretory pathway defects. D. Peroxisomal syncytia. Answer key, B. Explanation. Tay-Sachs disease and Gaucher's disease are classic examples of lysosomal storage diseases, LSDs, where missing hydrolytic enzymes cause undigested materials to build up and damage the cell. 6. The face of the Golgi apparatus that is closely associated with the endoplasmic reticulum and receives incoming transport vesicles is called the A. Trans face. B. Cis face. Correct. C. Median cisternae. D. Secretory face. Answer key, B. Explanation. The cis face, also called the forming face, is oriented toward the ER to receive raw proteins. The trans face, maturing/shipping face, points toward the plasma membrane to release finished secretory vesicles. 7. Which cytoskeletal filament is composed of tubulin dimers and forms the structural framework of cilia, flagella, and the mitotic spindle? A. Microfilaments. B. Intermediate filaments. C. Microtubules. Correct. D. Myosin filaments. Answer key, C. Explanation. Microtubules are hollow cylinders made of alpha and beta tubulin proteins. They are responsible for making up the spindle apparatus during mitosis, as well as the internal structure of cilia and flagella, 9+2 arrangement. 8. The inner fluid matrix of a chloroplast, which contains enzymes for the dark reaction, Calvin cycle, circular DNA and 70S ribosomes, is called the A. Cristae. B. Stroma. Correct. C. Matrix. D. Thylakoid lumen. Answer key, B. Explanation. The gel-like fluid inside the chloroplast is called the stroma. The term matrix refers specifically to the inner fluid of mitochondria, while cristae are the mitochondrial inner membrane folds. 9. The outer, relatively rigid, gel-like region of the cytoplasm located directly beneath the plasma membrane is known as the A. Endoplasm. B. Ectoplasm. Correct. C. Nucleoplasm. D. Cytosol. Answer key. B. Explanation. The cytoplasm is divided into two zones. The outer, clearer gel layer is the ectoplasm, or cell cortex, and the inner, more fluid sol layer containing the organelles is the endoplasm. 10. Which non-membranous, dark staining region within the nucleus is responsible for the synthesis of rRNA and assembly of ribosomal subunits? A. Chromatin. B. Nucleolus, correct. C. Nuclear pore. D. Nuclear lamina. Answer key. B. Explanation. The nucleolus is a dense structure inside the nucleus where ribosomal RNA, rRNA, is transcribed and combined with proteins to form ribosomal subunits before they exit to the cytoplasm.